Thanks, David and Han, for the invitation. It's very nice to have the opportunity to present this work here. First, I want to acknowledge that this is the work of Joan Gort, who's in this picture, who's a brilliant graduate student that just started in the lab. And yeah, the title of the talk today is Flexible Analog Computation in Low-Rank Balanced Spiking Networks. So we're going to be talking about analog computation. And so I guess, yeah, we all know that even though actually neurons communicate through discrete action potentials, computation is generally assumed to involve manipulation of analog variables. And so when we model spiking neural networks using mean field theories, implicitly we're assuming that the relevant object of computations are analog, are firing rates. And also when we use actually networks of firing rate units, there we explicitly assume that neurons are analog. So there are two kind of main frameworks for thinking about analog computation in the brain. The first one, the excitation-inhibition balance, focuses actually on the emergence of firing rates or probabilistic activity in spiking neurons. And so, as is well known, in these kinds of networks, strong heterogeneous connectivity leads to a cancellation between the large mean excitation and inhibition received by neurons, such that the remainder of this cancellation leaves the membrane potential at a distance from threshold, which is of the same order of the standard deviation of the temporal fluctuations, and this leads to a well-defined notion of firing rate or probability of spiking from each neuron. And this these kinds of models have been very successful at describing a number of biological observations about the statistical structure of spiking in cortical circuits, as well as some functional properties as well. The other framework assumes neurons are firing rate models and has instead focused not so much on the emergence of probabilistic continuous spiking, but more on the kinds of things that one can do with firing rates. In principle, general purpose flexible computation. And so these are basically RNNs, and I'm highlighting here both kind of task-trained RNNs, where one uses machine learning tools to get the network to perform a particular task, or quite related or equivalent low-rank RNNs, where one assumes a particular form of the connectivity. And so these networks generate neural implementations of dynamical systems that can implement arbitrary tasks and show a good dynamical equivalence with experiments. However, there is a lack of kind of integration between these two approaches. So on the one hand, in general, excitation-inhibition balance provides too rigid constraints for population-level firing rates in large network and is not ideal for general-purpose computation. And at the same time, rate-based RNNs generically do not address a number of important issues. I guess first and foremost, the existence of spikes, then a number of bottom-up constraints, such as the range and distribution of firing rates, of course, firing rate statistics, or spiking statistics, because there are no spikes, or separation between excitation and inhibition, and possible functional consequences of kind of the digital to analog conversion that is produced at this site. And so our goal in this project is actually to overcome this limitation and provide a unified framework for flexible general purpose analog computation in networks of spiking neurons. So we're not the first people who try to address this. I'm highlighting here a couple of papers from the groups of Larry Abbott and Mark Churchland and Serjan Ostojic. But both of these investigations were mainly based on numerical analysis of network models. And so what we have done is to actually develop a theory of activity in balanced networks with a specific connectivity structure that allows them to produce flexible computation, as I'm going to describe right now. Okay, so the network that we use starts as a standard balanced network with an excitatory and an inhibitory population driven by excitatory external input. The connectivity in the network has two components. One is the standard strong connectivity from the balanced regime, which we write as a sparse matrix multiplying a matrix that defines excitation and inhibition. And then we perturb this with a low rank perturbation. And I call it perturbation because these connections are weak, whereas these connections are strong. Note that, in fact, this is also a low rank structure, in fact, a rank 2 structure. But for ease of presentation, I'm going to generally refer to this as kind of the balance subspace and this as the low rank subspace, even though they're both low rank. And also I can highlight that through this construction, this network is actually not, neither of them is formally low rank because this matrix is not low rank. And also that it respects Dale's law, so because this term is weak, the separation between excitation and inhibition afforded by this is maintained. Okay, so then we will consider networks of leaky integrate and fire neurons with two sources of input. The first one is I call I fast. So these are fast currents, and they are the standard connectivity in a balanced network, which has synapses that are of the order of 1 over square root of k, where k is the mean number of connections per neuron, and also a strong excitatory synaptic input. And we're going to perturb this in two ways. At the level of recurrent activity, again, we're going to add this term here, which is just a sum of low rank terms. And then we're also going to allow low rank perturbation of the external input. And for reasons that will become clear, the time constant of this connectivity has to be significantly slower than the membrane time constant. And in fact, the time constant of the fast synapses we're going to approximate to be 0, so we're going to be dealing directly with spikes. And so we're going to take this setup and then define latent variables as, on the one hand, the projection of the membrane potentials on the column space of the low rank connectivity. So these B's here, and then the spatial averages for the excitatory and inhibitory populations, which in fact, again, are the column space of the balance subspace. And I guess just, again, notationally, there are various conventions for these vectors, I guess M and N or U and V. I'm sorry, but I use a new one. So B is going to define the column space and A is going to define the row space of the low rank components. And so then these latent variables, together with the spatial variance of the firing rates, are the macroscopic variables that completely specify the dynamics in this network. And we're going to develop a mean field theory to compute the asymptotic value of these quantities as well as their finite size corrections, which we will compare with with numerical simulations. Okay, so I guess the first thing is that we obviously one can choose parameters so that the baseline behavior of this network corresponds to an asynchronous irregular state. So you can see here the raster of excitatory and inhibitory cells, the cancellation between the large tonic excitation and inhibition, broad distributions of firing rates, and irregular activity. Okay, and so on top of this, for the first part of the talk, I'm going to study a network that actually has no low-rank recurrent input, but instead has a rank 2 weak external sinusoidal input. So this input can be described as a linear combination, a sinusoidal linear combination of these two vectors, which are positive because we want to respect that the external input is excitatory. And so I guess the first thing is we can average across neurons and define this term, which is positive, and the fluctuations around this term through these vectors be perpendicular that give rise to these two terms, okay? This mean uniform term across all neurons, and then these spatial fluctuations. And so because this is a weak external input, which is of order 1, then this term here corresponds as subdominant perturbation on the large external input that defines the balance state, okay? And so this is going to lead in general to, again, subdominant change in the overall firing rates of the activity of the network. But the interesting term is this one. So again, these two vectors now by definition have zero mean and some variance, which I'm defining as 1 here, but it doesn't matter. And then in principle, they can be correlated, and I'm going to refer to their correlation as rho. So how does it look for this particular example with sinusoidal modulation? If rho is 0, then what I'm showing here is the input for 1,000 neurons. And you can see that it has, as it should, 0 mean and a random distribution of phase and amplitude across all cells. And so that means that this input here is not adding any net input. The net input has been collected into this term, and is simply adding spatial variability. We can also compute the mean squared of the magnitude of this term, okay? And that's what I'm plotting here. So this is the squared input, and this is its temporal average. When the vectors are uncorrelated, then the temporal average is constant. But when the vectors are correlated, then that means that neurons develop an inhomogeneous phase preference, and this leads to a time-varying change in the magnitude of the latent state. So I'm going to be referring to this throughout the talk. This is an important quantity, the norm of the latent state, and that's what's being shown here as time-varying whenever there is an overlap between these two vectors here, okay? And so in general, the norm of the latent state is just going to be two coefficients that describe the coordinates of the external input in an orthogonal basis in the column space generated by the weak external input. Okay, so then what happens when we feed this kind of activity to the network? We can also plot the activity like this. So this is one factor, and this is the other factor, and it's an ellipse instead of a circle because I'm going to be focusing on this case here where there is some correlation between the two vectors such that the norm of the latent state is time varying, okay? And so that means that there's an ellipse, and I'm gonna use a color code in this plot, so that dark means moments where the norm of the latent state is larger, and light means the norm of the latent state is smaller. Okay, so if we drive the network with this input, we see a sinusoidal modulation in the activity of the cells. If we focus on a short window of time, a window such that the latent state can approximately be considered constant, then of course there is a well-defined notion of firing rate on this time scale. Okay, and we are going to use this, or in general, this allows one to define an instantaneous tuning curve relating the probability of spiking of each neuron, which we can compute, to the instantaneous value of the latent variable through the mean field theory. And so one can write this like this: the instantaneous firing rate of cell I for a given latent state V, in this case, because these are integrate and fire neurons, is the Ricciardi nonlinearity, which depends on the mean and the temporal variance of the voltage, and which are both functions of the latent state. And in addition, the mean varies from cell to cell. Okay, so let's look a bit more carefully at the behavior of this network. So what I'm plotting here is, in this curve, the mean membrane potential across neurons in the network. And again, I'm color coding it according to the norm of the latent state. This is the firing threshold. And so you see that there are fluctuations of order 1 of the mean depolarization across neurons. And what I'm plotting here is the firing rate, the mean firing rate of the network, which seems to also oscillate coupled to the norm of the latent state. Now, again, all we are changing is the weak external synaptic input. The large external input excitatory, which defines the balance state, is constant. And that means, I should be familiar to those of you familiar with the balanced formalism, that actually firing rates should be constant. And that is indeed what we see. This is a finite size fluctuation. So if we compute the autocorrelation of the firing rates as a function of the number of connections per neuron, we see that they decrease, and eventually asymptotically they vanish, as we expect from a balanced network. However, the autocorrelations in mean membrane potential actually increase with the network size. And that means that first, since the large spatially uniform external drive is constant, as I said, the mean firing rate in the balanced network is asymptotically constant too. But this constant global firing rate coexists with large, globally synchronized subthreshold membrane potential fluctuations, which track the norm of the latent state. So how can this be happening? How can we have constant firing rate on top of overall synchronization of the membrane potential? So we can gain some insight by focusing, instead of just on the mean membrane potential, on the spatial distribution of membrane potentials. And that's what's plotted here. And so we see that the standard deviation of the membrane potential across neurons also fluctuates, coupled to the norm of the latent state, but in antiphase. So the periods where the network is more depolarized, the mean membrane potential across neurons is broader. So spatial fluctuations in VM also coherently vary in antiphase with the mean membrane potential in an attempt to restore balance at the level of the mean firing rates. So our mean field theory predicts well these properties both of the mean firing rates n of the subthreshold membrane potentials. We can describe this both asymptotically and for finite sizes. You can see here is quite accurate even for numbers of connections which are relatively small. So, of course, these variations in membrane potential across neurons have implications for the distribution of firing rates in the network. And you can see that here. So here I'm plotting a raster plot. Down here, you're seeing the standard deviation of the distribution of firing rates and its prediction from the theory. And you see that even though the network is keeping the firing rate constant, this is done through modulation of the width of the firing rate distribution that tracks the norm of the latent state. Okay, so what's going on? How is this happening? So I'm gonna show you schematically what's happening in a plot like this. So on the x-axis, I'm plotting the distribution across neurons of the synaptic input or mean membrane potential. And we can go from this quantity to firing rates by passing the current through an expansive nonlinearity, obtaining the well-known right-skewed distribution of firing rates of the balance state, which will lead, for this particular situation, to a mean firing rate across the neurons. So now, as we have just said, when the norm of the latent state increases, this is going to lead to an increase of order 1 in the variance across neurons of the membrane potential, and this is going to lead everything else kept equal to a broadening of the firing rate distribution, which of course implies higher mean firing rates. But if this is the case, this would break the balance because the external input is clamping the firing rate of the neurons to a value, and the large external excitatory input hasn't changed. So in order to prevent the breaking of the balance, what happens is that the mean firing rates slightly adjust, so subdominantly, order 1 over square root of k, resulting in a global mean hyperpolarization, which is of order 1, and that you can see here from the shifting of this distribution leftward. And the shift is such that it cancels the increase in firing rate due to the broadening of the distribution. Okay? And so balance is preserved through a novel cancellation between the mean and the variance across neurons of the membrane potential, which tracks the norm of the latent state. So what are the implications of this? So again, if we can look at this linear manifold that we obtain by projecting the membrane potential on these two vectors. But we note that the globally coherent mean membrane potential mode tracks the modulo squared of the latent state and thus repeats at twice the frequency in this particular example where the input is sinusoidal, distorting the linear manifold. And so we obtain a picture like this. This plot on the left is what you would obtain taking this plot on the right and projecting it onto the xy axis. So you get the first latent variable and the second latent variable. However, the mean membrane potential oscillates at double the frequency, generating this curvature of the manifold. This is, again, a novel feature of our theory, as in standard low-rank theory, the manifold of free activations or membrane potentials is linear. So can we find evidence for this type of nonlinear manifold distortion? So we looked for it on experiments that use sinusoidal input. And in particular, we looked at the results in this paper from the lab of Ken Harris and Matteo Carandini. The purpose of the paper is a bit different from us. They want to explain how high-dimensional activity at the level of firing rates is consistent with low-dimensional activity at the level of pre-activations. But nevertheless, there are some interesting observations that bear upon the question that we are studying. And so basically the data set that we're going to be using, or the results that we're going to be focusing on, use oriented ratings, many orientations, and are 2-photon recordings from V1 and surrounding areas using calcium imaging, which is deconvolved for a total of around 20,000 neurons. And so in the paper, they develop a method for dimensionality reduction, which instead of being a standard autoencoder has an input part, which is a normal multi-layer perceptron. So that's standard, and that is meant to recover the pre-activation low-dimensional latent variables from firing rates. But then as the expansive term, they use actually a composition between a linear term and then a nonlinear term given by the static nonlinearity of the cells, like this. And so this is interesting because this part here is similar to kind of low-rank networks or to the network we are describing. And so let's see what they find. So when they drive the network with oriented ratings and then use this method to infer low-dimensional latent variables, they find that the dimensionality of the data is equal to 3. And these 3 latent factors are shown here. And then this is the activity of the cells. Note that the first 2 correspond to the gratings, of course. So they have the same frequency as the orientation frequency of the grating, okay? But there is this third term that, if you look closely, wiggles faster. And so when we plot our data in this way, we obtain something that is remarkably similar. I emphasize that this plot doesn't involve any tuning of parameters. It's simply the data that you've been seeing so far. So these 3 curves here are the first 3 principal components of the membrane potentials that I've been showing you. And so the interesting latent is the third one, right? So it has this frequency doubling that I've been describing to you, and it bears a particular phase relationship with the first 2. In particular, the troughs of the third latent align with the peaks and troughs of the first latent dimension, and the peaks of the third latent align with the peaks and troughs of the second latent dimension. And so this is a robust prediction from the model that I have described to you, and which seems to hold at least for this data set. So this prediction about the curvature of the low-dimensional manifold is consistent with the structure geometry of latent activity in mouse visual cortex. Okay, and then to conclude, I will describe another example where actually now we do use a low-rank perturbation to the recurrent connectivity. And so in this example, we're going to try to obtain a simple bistable switch, okay? And so we chose this particular example because historically this has been a problem where people have studied it both with functional motivation of obtaining multi-stability or bistability, but also where there has been quite a lot of work trying to obtain this functionality in the context of physiological properties that are consistent with observations in the data, in particular low firing rates in the elevated activity state, irregular firing, etc. And so this is a good example of a problem where there are both bottom-up and functional constraints that we can study. And so our approach again is going to be to add a sparsified rank 1 perturbation to the connectivity of the balance state. We're going to let the column of this rank 1 perturbation be a random vector, and we're going to train the elements of the row space to obtain the desired dynamics. So I guess I didn't have time to derive this equation, but it's a standard equation in these kinds of models which seek to approximate a dynamical system through functional approximation. So you have here the dynamics for the, in this case, single latent factor, and they are going to be given by a linear combination of the tuning curves of the neurons as a function of the latent state. And we train these A's here so that the overall dynamics has this shape, okay, which is kind of the canonical shape for a 1-bit bistable switch. And so when we do it, then it works. So you can see here in black the external input, and when we switch from positive to negative, the system alternates between these two states. In this case, we chose the two fixed points to correspond to approximately symmetric values of 1 and -1 for the latent variable, but I guess one can study this problem in any other condition as well. So how is the network doing this? So again, here is the target, and we can look at the values of the connectivity once the network has learned to perform this task. And so here I'm plotting each dot is a neuron, the value of the column space, and the value of the row space. And what we see is that the network finds this solution where there are 3 clusters, which we have colored in green, black, and blue. And each of these clusters contributes. Actually, the black cluster, because the A values here are close to 0, almost doesn't contribute. So the network selects 2 groups to perform the task. And if we linearly project the tuning curves of these cells or of these cells, we obtain this curve and this curve, which when summed give us the desired function. So we can look a bit deeper. If we just focus on the green wing, the other one is symmetric, so we're just gonna focus on this one, and we color code the neurons and look at their tuning curves. So these neurons here are the yellow, and then as we go up, it becomes darker. You see that in this case, there are unimodal tuning curves that vary a little bit in the slope of the relationship between firing rate and the latent state, and a little bit on the offset or threshold. And so now if we linearly combine them according to these the A's for each of the groups, then we see how it works. Basically, what the network is doing, it's flipping the sign of the tuning curves basically according to their threshold. And so that way it can get this non-monotonic shape, which produces the left wing, and the same thing is happening for the right wing. These black neurons, even though they don't really contribute much to these dynamics, they're interesting because when we look at their tuning curves, the tuning curves themselves are non-monotonic. And this is another thing that is novel about our framework, that it produces, through the nonlinearity that I've described, non-monotonic tuning curves as a function of the latent state. In this particular problem, that's actually not very useful, but this is something that is both useful in general because it complexifies the basically repertoire of the network for modeling different dynamics because it has tuning curves for individual cells that are more diverse, and also is experimentally observed where one typically sees complex nonlinear dependencies of the firing rate on the latent states. And then I guess it's interesting if you now take the activity of the neurons and then you plot them on top of the FI curve. And you see, as you expect, that actually for both attractors, the activity of the cells lives here below threshold. And so this shows that, of course, even though it's important that the neurons have a nonlinearity in terms of shaping these tuning curves to the latent state, which eventually allow the system to produce the kind of nonlinear dynamics that it needs to do working memory. There is no particular role of the overall firing rate of the network or the spiking threshold, which were important quantities in kind of the models for obtaining low rate irregular spiking activity in the past. Okay, and so with this I'll summarize. So we developed an accurate mean field theory of low-dimensional dynamics in balanced networks with low-rank perturbations to their connectivity. The theory starts from the realization that dynamics in the balanced subspace and in the low-rank subspace are coupled. Approximate low-rank perturbations to the connectivity of a balanced network will tend to disrupt balance through an increase in membrane potential fluctuations, which are driven by changes in the norm of the latent state. Firing rates will adjust to their perturbation subdominantly, so they will change by order 1 over square root of k, their activity, in such a way as to drive a net hyperpolarization that restores the balance, but leaves a signature at the level of the relationship between the norm of the latent state and the shape of the distribution of firing rates. So this is a form of coupling from the low-rank subspace to the balance subspace. Coupling in the other direction depends on the shape of the row space of the low-rank connectivity, which I haven't explored very deeply today. This new form of balance curves the low-dimensional manifold where the dynamics takes place, and we provided some evidence of this nonlinear behavior from recordings of mouse visual cortex. And then in principle, any form of continuous dynamics can be produced by the spiking network, and we studied an example of a network implementing a 1-bit working memory. Overall, these results demonstrate the feasibility of flexible, arbitrary analog computation in networks of spiking neurons, which obey well-established anatomical and physiological constraints. Okay, and then with this, I will acknowledge Joan again, who developed the theory, the other members of our lab, our collaborators, and funding sources.